Click the bell icon and subscribe to our channel so that you will not miss any classes. In this class, let us discuss the chapter Cell Cycle and Cell Division of the Unit 3 Cell Structure and Function. This chapter, Cell Cycle and Cell Division, is found to be very important chapter for next year. That means in second PUC, there are concepts like genetics and molecular biology. In order to understand those particular topics, for example, structure of chromosomes, how they divide, chromatin, the concepts like that, you have to understand this chapter and the concepts present in this chapter in detail. So on that course, this chapter is found to be very important. Now, let us discuss first what is meant by cell division. All living organisms, as we know, they are made up of a cell. The cell will divide. The cell will divide to produce two identical daughter cells. If we consider this as a parental cell, the parental cell will divide. Meanwhile, it will grow also. It will grow and finally it will divide to produce the daughter cells. And this generation, this will constitute the generation. That is, we consider it as first generation. And these cells will also grow in their size. We know that growth in case of cells is considered as increase in their cytoplasmic content. And finally, they will also divide, and this will constitute the second generation. The process will continuously happen, and finally it will result in million number of cells, which will constitute one particular structure, and that particular structure could be organ, or it could be an organ system, or it could be an organism. So, the cell division is defined as the process of formation of identical daughter cells from a parental cell. Parental cell is called cell division. The process of cell division was first demonstrated by two scientists, Boveri and Fleming. In the year 1879. They detailedly studied the process of cell division in different types of cells, and they given a final structure or final thing, that is the process of formation of identical daughter cells from a parent cell is called the cell division. In order to understand in detail the process of cell division, firstly we need to understand the cell cycle. Cell cycle. Till now we know that during cell division the cell will divide into two daughter cells. Now during cell division, cell will not only divide but also it will grow in its size. The growth of the cell will take place. Meanwhile, there is a replication of DNA takes place. The DNA replication will also take place, and all these process, division of the cell, growth of the cell and replication of DNA present in a cell should take place in a systematic way. In a systematic way, and these processes will togetherly constitute the cell cycle. That means the series of cyclic events, we can say. It is a cyclic process. Series of cyclic events which takes place during the formation of daughter cells from a parental cell is called cell cycle. That means the series of events means these major events are division of the cell, growth of the cell, and replication of the cells. They should take place in a systematic way during the division of the cell, and these events will constitute the cell cycle. That means the definition of cell cycle is the series of cyclic events which are taking place during the process of formation of the daughter cells from a parental cell, that is during cell divisions. If we consider this as a parental cell, initially the parental cell will grow in its size and finally it will divide and produces two daughter cells. So if we consider this as a cell cycle, during this from the parental cell there is formation of two daughter cells, and from these daughter cells again they grow in their size and again they will divide and produces two daughter cells. That means it is a cyclic process. So all the events during this, it could be any type of the cells, all the events will be the same. But the thing is, there is a time required for this. During the formation of daughter cells from a parental cell, there is some time required, and this time is called a generation time. That means the time required to produce two daughter cells from a parental cell is called a generation time. That means that generation time will differ in different types of the cells. Say for example, the generation time of a bacterial cell. If we take a bacterial cell, the bacterial cell will take only 30 minutes to divide and produce two daughter cells. And yeast cells, they require 90 minutes to divide. And the myeloma cells of intestine, they require 8 to 10 hours to divide. Whereas human cells in a culture, that means if we take cells from us, if we take the human cells out and if we grow it in a laboratory condition, so that is, we call it as in culture. So when we grow the human cells in a laboratory condition, there they will take approximately 24 hours to divide. So by taking this as a reference, so the scientists have told that approximately in all eukaryotic cells, the time required, that is the generation time, is found to be 24 hours. That means in all eukaryotic cells, the generation time is 24 hours, but in prokaryotes it will differ. For example, in yeast it is 90 minutes and in case of bacteria it is 30 minutes. And in different types of bacteria it will also divide. That is not of concern here. We shall discuss in the chapters wherever it is applicable. the cell cycle is majorly divided into two phases. One is interphase. It is also referred as the resting phase. Interphase or the resting phase. Again, this interphase is divided into G1 phase, or we can call it as gap 1, S phase, or it is also called synthesis phase, and G2 phase, or we can call it as gap 2. This is gap 1 and this is gap 2. During these phases, there are particular processes which are taking place. We shall take them in detail. And the second phase is the M phase, or we can call it as division phase. That means during interphase, the cell is not dividing. It will be at rest. Even though we call it as resting phase, there are various events taking place. We shall discuss them here. But during M phase, the cell will not be in rest. It will be in a division condition. But the cell will spend more of its time, that is cell cycle will be having some time, right? So that time is majorly made up of the interphase. Say for example, we told that the generation time of eukaryotic cells or per typical human cell or typical eukaryotic cell is 24 hours. So in 24 hours, 95% of it will be spent in, 95% means approximately 23 hours will be spent in interphase, and the remaining 5%, that is in about 1 hour, will be spent in M phase. So it is true for all kinds of the cells. It could be eukaryotic or prokaryotic, anything. Here the 95% of the time, generation time, will be spent in interphase, and remaining 5% is spent in the M phase or the division phase. So now let us take these in detail. We shall draw one typical cell cycle. Say here is one cell cycle, and here the cell is. The cell initially it will grow in its size. Initially it will grow and it will complete its life cycle, and finally it will divide into two. This is one cell I am representing. Let me write one single here. So it will divide into two daughter cells. So here is a time spent. We can represent it as G1 phase, and here is S phase, and here is G2 phase, and here is the M phase. Here is the M phase, that is the division phase. So let me write this. So after M phase, immediately it will enter into the interphase. After completing M phase, immediately the cell will enter into the interphase, that is G1 phase. And in different cells, there are different conditions. We shall discuss in the exceptional cases further. And this constitutes the cell cycle. So if we see here, the cell will spend more of its time in the interphase. So let us take up the events which are taking place in the particular phases. So firstly, the G1 phase. In G1 phase, the cell is metabolically highly active, even though it is called resting phase. That is, it is a subphase of interphase, right? Interphase means resting phase, we told. But even though it is called resting phase, the cell is metabolically highly active. That means it will be synthesizing the structural and functional proteins required for cell division. That is, in M phase, there is a requirement of some structural and functional proteins, and those are synthesized in the M phase. Those are synthesized in the G1 phase. And the cell organelles like chloroplast, Golgi apparatus, endoplasmic reticulum, lysosomes, and the different types of RNAs. We know that there are 3 types of RNAs: mRNA, tRNA, and rRNA get synthesized. During the G1 phase, there is no replication of DNA taking place. The DNA replication will not take place. The cell will grow in its size. The cell growth will take place. That means increase in the cytoplasmic content will take place. And also there is production of some substances. There is production of some substances like protein kinases. So these were found to be important for the competitive exams. There is production of substances called protein kinases, which are important for further cell division. That means whether the cell has to enter into next phase or not will be controlled by the substances called protein kinases. And these protein kinases are controlled by, the protein kinases are controlled by proteins called cyclins. So you should remember here, whether the cell has to enter into S phase or not, or it should be presenting G1 phase itself, or it should continue. So that phenomenon is controlled by the substances called the protein kinases, and the protein kinases are under the control of the proteins called the cyclins. That is, it is a master, it is controlling these. So these are called as, that means the substances which are controlling the cell division are called cyclin dependent protein kinases. Cyclin dependent protein kinases. Cyclin dependent protein kinases will regulate the further division of the cells, and these will be produced in the G1 phase itself. And there are different kinds of cyclin dependent protein kinases which are produced at the different phases. For example, For controlling G1 phase, the cyclin dependent protein kinases are of different kind. They will be produced in the G1 phase and will be destroyed in the G1 phase itself. And there are different types of cyclin dependent protein kinases for S phase. They are produced in the S phase and they are destroyed in the S phase itself. That you should remember. That means cyclin dependent protein kinases are there in all the phases, but they are of different kinds and they are produced and destroyed at the particular phases. So they will regulate the process of cell division. So these are the major events which are taking place in G1 phase. That means gap 1. Here the cell will grow in its size and there is a increase in the cytoplasmic content, that is the growth. The cell will be metabolically very active and it will be involved in the production of structural and functional proteins, and also there is a synthesis of the cell organelles like chloroplast, endoplasmic reticulum, Golgi apparatus, lysosomes and all will be produced. Okay, so that is about the G1 phase.